Great, thank you so much. We have our last talk of today by Dr. Pulak from Union Biometrica, and the talk title is High-throughput imaging and sorting of organoids, spheroids, and cell clusters.
Okay. My name is Pulak, and I'm the scientific director at Union Biometrica. And today I'm going to tell you about our instrumentation for analyzing and sorting organoids and spheroids. Let's see, there we go. Yesterday afternoon at this virtual seminar, Mike Stednisky from Phytonix put up a slide that said, "Make me care." So I sort of thought I would start off by stating where I'm trying to get to with this presentation. And that is that there's this instrumentation that provides automation to increase the throughput of data collection. And if we think about how things have changed in the lab over the years and take a look at some representative pictures maybe over the years, a long time ago, there really wasn't a lot on the benchtop. There were a lot of bottles and stains and dyes. And today's researchers are working at the bench, but they do have some tools available to them, like microfuges, centrifuges, maybe automated pipetters, maybe fluorimeter right at the bench, but they're still at the bench doing a lot of the tedious, time-consuming, labor-intensive, error-prone work. Now, more and more workstations like these are being developed in core lab facilities to do some of that tedious, time-consuming work. And flow cytometers, especially ones that, that can dispense, belong in that kind of a setting. And I'm going to tell you about our flow cytometer and how it can fit into that kind of a setting. And of course, all flow cytometers can fit into that kind of setting, especially if they're dispensers. But I'm going to tell you about ours because it's uniquely able to work with these spheroids and organoids. So the benefits of flow cytometry: it's a flow cytometry group. It's we all know, multiple measurements per object, integration, interrogation of every object in the sample, unbiased analysis. So take out some of that subjectivity that might be introduced by the researcher, large sample numbers so that you can look at small differences, and then the increased throughput of handling these very tiny objects that there really aren't a lot of way of accurately moving them around from place to place. Now, my focus, I'm going to focus on showing you examples of what we can do with cell clusters. And so cell clusters can be generated in a lot of different ways, and this slide sort of shows you 4 of those ways. Stem cell researchers especially work with Matrigel, where they can encapsulate cells within the Matrigel matrix and then submerge that in culture media where they can add certain transcription factors and certain cytokines to drive the differentiation of these stem cells along certain developmental pathways. And those will grow, divide, and form little organoids, little spheres that look and take on the characteristics of some of the tissue that the researchers are trying to replicate. Another way is generating cell clusters is a hanging drop method, where you have an apparatus and you can, with a drop of media and cells, form this drop where the cells collect and then form a spheroid. Other ways of generating cell clusters are these bioreactors, spinner flask bioreactors, and then finally using micromolds or microwells where cells are in a media distributed across the surface of something that has multiple little microcavities. Those cells drop to the bottom of the microcavity and form these cell clusters. So there's a whole bunch of different ways of generating these cell clusters. And then how do you analyze them? How can you do stuff with them? And that's where our flow cytometer can be beneficial to a researcher. On this slide, I'm showing a schematic on the right of how, of what happens or how the flow cytometer works. And the schematic is really set up for the COPA's vision. And that's the flow cytometer that has the camera that can capture an image of the object as it flows through the flow cell. Now, most of the examples I'm going to show you are for cell clusters, organoids, spheroids, and other kinds of cell clusters. But I wanted to remind everyone or tell everyone that we also can work with beads and pollen and seeds as a sample. Other sample types include small multicellular organisms like C. elegans, Drosophila, embryos, zebrafish embryos, and larvae, as well as certain other sample types that are too large or too fragile for traditional flow cytometers. And I'll say something more about that in a minute or so. So the sample enters the flow cell from the top and is carried along with sheath reagent so that it's hydrodynamically focused as it passes through the flow cell. And the flow cell can have any of three or four, but I'll tell you about 3 different widths. And the smallest is a 250 micron square passageway, microfluidics channel, so 250 microns across, or 500 microns, or 1,000 microns. So already you recognize that our smallest flow cell passage is at least 2, maybe 3 times wider than traditional flow cytometers. So it should be able to handle a much bigger sample type. So that's one point I wanted to make. Then I wanted to point out as the sample passes through the flow cell, it will go through either 1 set of lasers or 2 sets of lasers. Fluorescence data is collected orthogonally on PMTs. In line, we have a forward scatter detector as well as an extinction detector. The extinction detector is a measurement of how much light can pass through the object. That detector also provides a size measurement. Because we're working with objects that are so big, we can capture the length of time that a shadow is cast on that detector. And because we have stable laminar flow through the flow cell, that means constant velocity. Therefore, the length of time that the shadow is cast is proportional to the diameter of the object. So that's our measurement. Our measurement is not the scatter measurement, it is the length of time that the shadow is cast on that detector. So the sample continues through the flow cell and the camera is triggered from the flow cytometry data collected. So whatever triggers data collection up here can be used to trigger data collection of an image. So we can decide to collect images of everything going through the flow cell, and every time it gets triggered and flow cell data is collected, or we can draw a gate region and only collect images from the region of interest. Now, another difference between our flow cytometry, besides this wide bore flow channel, is our sorting mechanism. So normally the stream leaves the flow cell and is diverted to a waste or sample recovery. However, if an object passes and meets the sort criteria, then a signal is sent to the air diverter to turn off and then on again, and that generates a drop or a slice of liquid that can continue downward while the remaining stream is again diverted to waste or sample recovery. So the droplets that we're generating are not the tiny droplets that most traditional flow cytometers generate. We're generating a fairly large droplet that contains the object of interest. Now, I mentioned fragile sample types. Because of the larger flow diameter and the low pressures that we work at. So the COPAS vision, the highest pressure that the COPAS vision works at is 5 psi at the flow cell. So that's considerably less than traditional flow cytometers. This means the combination of the lower pressures and wide flow cell of microfluidics channel means that there are less shear forces and less damage imparted on the sample as it passes through. So most of what I'm going to tell you is going to be data, most of what I'm going to show you is data collected on the Copas Vision that has the imaging, and it's a flow cytometer that collects these images. However, I did want to mention that we do have other kinds of flow cytometers, Copas F and a biosorter. They do not have the imaging, but if you're interested in those, please go to our website. Okay. My first example of the kind of experiment that you can do with on a Copas Vision is shown here. And this is spheroids that are generated in Corning microcavity plates, and they're actually microcavity flasks. 2 flasks were set up where the density in one flask of starting cells, single cells, individual cells, was at a high density and therefore generated large spheroids because lots of cells fell into the microcavity well, and another flask where that was seated at a lower density so that fewer cells fell into each of the microcavities and smaller spheroids were generated. They were then mixed, and here's the results of the data from that mixture. We see small ones, we see larger things, and we see larger still, a few scattered larger still. We run these through and retrospectively can look at the images after we've run them through. We can look at them live as well, but they're going by so fast that you're only seeing an image every so often. So once the data is collected, you can retrospectively look at what fell into this region and what fell into this region and what's way over here. And we see that in this, the objects that by flow cytometry data are smaller and have less optical density are these smaller spheroids. Those that are in this region are bigger and are more opaque, and therefore increase in optical density, increase in extinction. We were curious about what was up here, and we draw a gate region around here and take a look at retrospectively look at what was up here. We see that there were these dimers and occasionally trimers and other larger kinds of objects that were up in this region. So having this capability, and once we know the region that we're after, and we could do that simply on the basis of size, but we could run a little bit of sample through, first determine where the objects are that we are most interested in, and then we could sort them into wells of multi-well plate. This is an image of an entire 96-well plate, and it shows that one spheroid, and certainly at way back here, these all look uniform in size. They're pretty close to being the same size this way, but this was from the flow cytometer region R3, and they are pretty similar in size. And they're similar in size because of the cavity that they came from. It takes about a minute to a minute and a half to fill a 96-well plate with one object. Another example I'd like to tell you about is work that we did with a researcher that's working with stem cells, trying to drive those stem cells to behave like the beta cells from pancreatic islets. And these are the cells that secrete insulin. So their setup is stem cells that are transformed with a GFP reporter that is expressed off of an insulin promoter. So if the transcription factors for insulin are there, GFP is going to be expressed. And so that's their assay. So they grow these stem cells under various conditions. These tend to form clusters in the spinner flasks, and now they want to analyze those clusters. And they're analyzing by measuring the GFP expression. So in this example, we ran some of the clusters through, we determined the fluorescence that are coming from these clusters, and now they can take a look to see if under certain conditions they're driving this more towards the right and up, or not more towards the up to the right and up. So to determine the conditions that are best for maximizing the insulin secretion, and then use those with non-transformed human cells with the long-term goal that these could be generated for transplantation experiments for people with type 1 diabetes. Long-term goal. They're not quite ready yet to do those kinds of experiments. These are experiments to take them there. Another example is tumor spheres, again, generated in a bioreactor. When we run a sample from the bioreactor, we see all kinds of different sizes and different autofluorescence in the green channel. These are non-transform, these do not express GFP. This is just the autofluorescence in the green channel. So you can see we're all over the place here. So again, if we run these through and then draw a gate region and retrospectively look at the images from each of these regions, we can see that down here we have near the small end and the low autofluorescence end, we have all kinds of things, little kinds of different small cell clusters, small single cells, small little particles. We have no idea what this is, but all kinds of different things. This region over here that looks maybe like the most concentrated, we have fairly uniform spheroids, all about the same size, all about the same optical density. And then some of these larger things look like maybe a couple of these joined together, maybe some kind of aberrant growth, maybe some that joined early and then started growing, but all kinds of different things going on here. But this would allow a researcher to grow up in a bioreactor, lots of these different things, and then sort out ones that are uniform size to then do an experiment where they're comparing the effect of drugs or growth conditions of tumor spheres of the same size, and be able to come up with a better answer than if you were simply distributing this in a random way into wells and multi-well plates and then testing it. So it makes the experiment a little more precise. And this is work that we did with our customer at Encapsulated at NIH. Another example that I want to point out is that there are some people that are trying to, the use of Matrigel is really big in the stem cell field, and it's very expensive and it's a bit cumbersome to work with. However, there are some people that are trying to generate hydrogel particles that encapsulate the cells and then allow the cells to differentiate the way they would when they're in Matrigel in a dish, and to grow them up in batch and to be able to distribute those then into wells and multi-well plates. So with our technology, you can sort out the empty Matrigel particles from those that have encapsulated a cell cluster. So this is another kind of an experiment that can be done with our technology. We've worked with Neurospheres. One of our customers in Germany has worked with human mouse and rat neurospheres. And again, these are cell clusters that can get to be pretty big, 500 microns in diameter. And mature neurospheres can be induced to differentiate all the different neuronal cell types. So the starting material for a bioreactor of neurospheres, again, has all kinds of different things present. We can run those and sort on the basis of size and optical density, sort out those that are larger, and generate a fairly uniform size of neurospheres for downstream experiments. Some simple downstream tests that were done was viability, as well as the determination if the neurospheres could differentiate into the different neuronal cell types. Both were found to be good. They do differentiate, and their viability is very similar. The last example I'm going to give you is with mouse retinal organoids. And on this slide, it will give me an opportunity to tell you about profiles. So we have a feature called a profiler that allows you to generate this kind of two-dimensional graph that shows the distribution of the signal along the length of the object as it goes through. this organoid down here, the blue line identifies the optical density, the extinction measurements. So as the edge comes in, less and less light is, or more and more light is blocked, and that signal increases. And then as the spheroid or organoid is leaving, it comes back down to baseline. This is not GFP. This is low-level autofluorescence in the green channel, most likely. Up here, we have an organoid that has cells expressing GFP off of a reporter, but only some of those cells are expressing GFP off of a reporter. So as this organoid went through, and this part of the organoid went through the blue laser, the GFP got excited, and we collected measurements from one part of that organoid. So this over here, we have the dot plot, and this is the region that we're only looking at objects that fell within this region. Over here is a picture, is a representation of the image that our bright field, that our camera collects, and with it overlaid is the profile. And it shows that while the image doesn't tell you there's GFP, the profile does tell you that. So here we have organoids that were dispensed into wells and multi-well plates, and just the images on the next slide, or, ooh, yeah, so this is on the next slide, I'm showing you the profiles from those organoids, and you see that rows A and B, the GFP, the green fluorescence level is very low. And that is because we selected a region that had, on our sorting window, that had low GFP, but fell within that, fell within the R3 region. So those things of this size, but low GFP, and then sorted those out in rows A and B. In rows C and D, we sorted out those with increased levels of GFP. And these are not our images from our COPAS vision. These are pictures off the microscope for the organoids that were dispensed into those wells. So you can see those with low green fluorescence have low green fluorescence when you look at them under a microscope, and their profiles have low green fluorescence. So it verifies that our profiler is giving you the answer that the microscope would, but it's doing it on the fly for every object that goes through, and can do this at tens per second. Those with higher levels of GFP, as we see it in the photomicrographs, also have higher levels of GFP in their profiles to support, and is supported by the photomicrographs. So this is work that was done with Peter Baranoff. They're trying to figure out the growth conditions to drive as many of these organoids as possible to express these retinal markers, so that they could possibly figure out a way of growing these organoids for the treatment of diabetic macular edema or other retinal disorders, and use those in transplantation. Again, very early stages, but this is how this technology can be used. The last thing I want to mention in the last minute here is that we have an autosampler to load samples from wells of multi-well plates to our flow cytometer. So you can probe will go into the well, draw the contents out, send it to the flow cytometer. The flow cytometer can analyze and then redispense into wells and multi-well plates for further analysis, for either assays or further analysis by microscopically or some other way, possibly sequencing, possibly determining transcription profiling or something else. So in summary, I've mostly told you about COPAS vision, but it's a large particle flow cytometer for analysis and sorting, and it can capture an image. We have 3 different size flow cells, 250, 500, 1,000 micron, 4 or 8 PMTs, up to 4 lasers. We capture a bright field image. we get the real-time profiling of the object and can use those parameters for sorting and dispensing based on size, optical density, scatter, and fluorescence, and gentle dispensing into wells of multi-well plates or other collection devices like tubes and dishes. Thank you for attending the presentation, and if you have any questions now, otherwise, I know there's a lot at our website, and please inspect that as well.
Thank you so much, Rock. That was an absolutely wonderful presentation, and I think that was the most perfect timing possible, right at 25 minutes, which leaves us 5 minutes for questions. So we do have a couple of questions that came through. And the first one from Ira is, what is the smallest diameter object that the Copus can resolve?
So the smallest size depends on what it is. We have run beads that are 2 micron diameter, and we're able to detect those and detect rainbow beads of various fluorescence intensities at the 2 micron level. However, cells are not beads, and we don't know that we're capturing every normal eukaryotic mammalian cell. It's because some cells are pretty small and they're more translucent than the beads are, and so it's difficult to really hammer it down and pinpoint it to something specific. But I like to say, well, down to 2 microns, because we do know that we can detect 2 micron beads. This is not, I often will say at a presentation, this does not substitute for your conventional, traditional flow cytometer. Those flow cytometers are great, all of them, they're great, they're wonderful. We don't really compete with the single cell flow cytometers. Our wheelhouse, if you want to use that term, is the larger objects.
Okay, great. Another question from Ira is, can you sort clusters based on the shape of the fluorescence over time profile?
Sort of, yes. So the shape it can be, because they're round, it's more difficult when something is round because of all the different orientations they can go through. But you can select something where there's fluorescence near the surface because there are peaks near the edges, and you can subparse your profile to be just the edges or to be more the center, and use that. Other shape parameters are, at this point, are a little too difficult because of the speed that things are going through. We are working at trying to develop some software that captures some of that information, so that we can say something about how smooth the surface is or how rough the surface is, and use that as a sorting criteria. I don't think we're quite there yet to use that broadly as a sorting criteria, but we are trying to work on other things other than just the flow cytometry data. Somehow capture some of that information out of the image.
Perfect. And we have 2 more questions in a minute and a half, so let's try and get through both. The first question from Rohit is, can we do the cytotoxicity assay in the spheroid models using the cytometer? And then they asked what channels and fluorochromes it supports. And I think you addressed that earlier, but if you want to just review that for them.
Yes, so you can have up to 4 lasers. So any of the dyes and stains that you would like to use, pretty much over the spectrum of the 405 to 630 laser excitation capabilities, and then the up to 8 channels, and those are, you can decide on which parts of the spectrum are most important to you, and with bandpass filters set up to collect that part of the spectrum. So yes, any of your live dead stains that you are currently using, you can customized to the extent to be able to fall within those 8 channels and those 4 lasers, and that almost covers most of what you can do.
Perfect. And then about 15 seconds for the last one. It's a very simple question, but a great question. Does the BioSorter and Vision systems, or do they have temperature control availability for the sample?
They don't, and they do have the aerosol containment, and they're in a box, so they've got some of that capability, but we do not have temperature, any type of temperature control on this.
Okay, that was perfect timing for all of our questions and for your presentation. So thank you again so much, Rock, for a really wonderful talk today. We appreciate it.
Thank you. It was my pleasure. Thanks.